Hey everyone, my name is Anaya Miller, and today I'll be presenting a recent study published in the Cell. It's titled Infant Microbes and Metabolites Point to Childhood Neurodevelopmental Disorders. It is by Professor Ahrens, actually, so shout out to her and colleagues. So this paper stood out to me because it explores how early gut life microbiome and metabolite patterns may help us better understand and also potentially predict neurodevelopmental outcomes in children. These outcomes include conditions like ADHD, autism spectrum disorder, and also other intellectual and communication disorders. So I thought the study would be a great fit because I like that it brings together many themes from infant microbial succession to metabolomics and gut brain axis communication. Also, shout out to our professor again, as she is one of the contributing authors. So let's get into it. So, before we dive into the content, I just want to break a few things down and walk through some common acronyms that may appear throughout the paper. Many of these are central to the microbiome research and neurodevelopmental studies. For example, ND refers broadly to neurodevelopmental disorders like ASD, which is autism spectrum, ADHD, and also there's terms like HLA, CFAs, and PFDA. These refers to genetic markers, gut metabolites, and environmental toxins. So knowing these upfront will help make the later slides much easier to follow along with. I'll just pause here for a few seconds. You can take a picture here, or you can just refer back to this point if you need to use it. They're pretty much words we are used to with the microbiome. So here's my bluff, the bottom line up front of this study. So researchers basically followed over 16,000 children from birth as a part of this ABIS cohort, and they discovered that the infants who were later diagnosed with neurodevelopmental disorders like ASD, ADHD, intellectual disability, and speech disorders, they had already shown measurable differences in their gut microbiome and metabolite profiles during the first year of life. So this is big. So for example, infants who developed NDs had lower levels of beneficial microbes like the Coprococcus, Akkermansia, and other ones like Roseburia hominis. So these microbes are known to produce anti-inflammatory compounds. This support gut barrier function and also influence neurotransmitter pathways like the GABA and serotonin pathways. So with this depletion, it suggests that a gut environment more prone to inflammation and immune dysregulation, which both have been linked to ND development. So dysregulation is what we see in multiple studies that are linked to ND development. So at the same time, we're looking at cord blood metabolomics. This revealed that future ASD cases had lower levels of linolenic acid, which is a precursor to anti-inflammatory omega-3 fatty acids, and higher levels of PFDA. This is a toxin associated with immune dysfunction. So these early life signatures were not only associated with microbiome changes, but also with other clinical factors like otitis, frequent antibiotic use, parental smoking, so environmental issues, and even HLA genotypes such as DR4 and DQ8. So this is tied to autoimmune risk as well. Overall, the study makes a strong case that multi-omic data, which is combining stool microbiota, serum metabolites, and early life exposures, could serve as early indicators of neurodevelopmental risk. So, what's especially important is that these differences were observed years before diagnosis, suggesting a window for possible early intervention or prevention strategies in high risk infants. So, to briefly put things into context, neurodevelopmental disorders like autism and ADHD, they're becoming more prevalent, affecting about 1 in 6 children. So while previous research has shown that gut microbiome can influence brain development, most of that work has been done in older kids after diagnosis. So this leaves a big unanswered question. Do these changes actually start in infancy? Answering that could help us identify early warning signs and maybe even intervene before symptoms begin, which is why this study is very important. So, I wanted to pull a figure to help us out a little bit with my background. So, this figure from a national survey of over 12,000 children illustrates the broad scope and associated factors of neurodevelopmental disorders in the US. So, here you can see that ADHD and ASD are the most commonly reported NDs, with nearly 1,100 and 379 cases, respectively, in this subset. So, learning disabilities and intellectual disabilities also appear in meaningful numbers. What's striking is the number of co occurring stressors reported, such as family addiction, incarceration, mental illness, or a child being bullied, all of which are known to affect how the brain develops in on one's behavior. So, these social and environmental stressors often intersect with biological risk factors, which makes it hard to isolate contributing ND risks and highlights why large comprehensive studies like the one in the cell are so valuable. So, why does this even matter? So, neurodevelopmental disorders represent one of the fastest growing pediatric health concerns, yet we still lack reliable early indicators to identify at risk infants before behavioral symptoms appear. Normally, one would take their child to see a specific doctor once they start showing traits and signs of ADHD or ASD. But see, with this study, we're trying to have an early detection system from infancy to where we prevent these symptoms from even showing. So, first, the public health burden is significant. Recent CDC estimates show that over 17% of the US children are affected by conditions like ADHD and other NDs. So, the prevalence yet has increased over time, yet early identification remains challenging. Second, there's a strong biological rationale for examining the gut microbiome. The infant gut is a hub of immune education, metabolic signaling, and neural crosstalk, all of which are essential for brain development. Multiple studies have proposed that gut dysbiosis may contribute to ND risk, but data in infants remain limited. Third, there's a critical diagnostic gap. Most children who are not formally diagnosed till preschool or beyond, and a few existing studies follow infants over time with clinically verified ND outcomes. So this is where our study actually offers a major advancement by combining multi-omic profiling with long-term diagnostic follow-up in large cohorts. So now this slide is going to break down the multi-omic methods used in this study. So one of its biggest strengths. So what's unique here is that the author didn't just look at the behavior or clinical notes. They actually took a deep biological dive across multi-infant sample types. So, starting with the cord blood, which was collected at birth, the researchers performed lipodomics, including LCMS. So that's basically liquid chromatography with tandem mass spectrometry. This technique allowed them to measure a wide range of lipids that reflect immune system priming, inflammation, and metabolic tone. So all of these are thought to influence NDs. Infant stool samples also were collected around 12 months of age, and this used a process called 16S RNA sequencing, specifically due to QIIME2 and DADA2. So these pipelines are standard in microbiome studies and allow for like high resolution profiling of the gut bacterial community. This study also incorporated genetic risk data which was sourced by genotyping HLA alleles, and it focused on those that are linked to immune-related traits that could modify neurodevelopmental vulnerability. Also, beneath the table, I wanted to highlight two key takeaways. So first, this study captures a diverse set of infant biosignatures. What's important to remember is that all the samples that we collected before neurodevelopmental diagnosis occurred, and also gives the study a real predictive value because these were collected before diagnosis occurred. So most diagnoses, like I said before, they occur after symptoms begin to really be are displayed. This is collected before we can, infer symptoms were shown or presented. So, by analyzing cord blood, stool, and genetic material, the researchers captured a multidimensional snapshot of early life biology. So, this may offer clues for early risk identification. Also, I wanted to point out that their methods were rigorous and reproducible. They didn't just rely on one platform or one analysis tool. They used a combination of different tools like DSQ, and they found basically that there were different abundant microbes for multivariable association testing. And also there are random forest methods to explore predictive patterns. Together these methods made the findings strategically sound and together these methods made the findings statistically sound and biologically meaningful. Overall, this multi design gave the study a power to move beyond correlation and start mapping the actual biological signatures of neurodevelopmental risk in infancy. So, moving along, this figure gives a full view of the study's population and how diagnoses were tracked over time. This data comes from the ABIS cohort, which stands for All Babies in Southeast Sweden. And it's a huge population based study that enrolls over 14,800 children who were born between the years 1997 and 1999. So, what makes the ABIS so valuable is that it's not just big, it's longitudinal, meaning that these kids were followed for decades. The researchers had access to not only just clinical outcomes, but also biobank samples like cord blood and stool from the infancy, which actually is rare in the study. That's why, as I was reading into this study, I was like, whoa, how did they have access to all this information? Panel A, we're looking at how many children were diagnosed with various neurodevelopmental conditions. This includes autism, ADHD, and most of our common NDs we noted before. So you'll notice that there's a large control group, over 14,000 children without an ND diagnosis, which gives the study a solid statistical power. One thing to pay attention to here is though the age of diagnosis, for example. So speech diagnoses were around 4 to 5 years old, and ASD and ADHD were diagnosed much later, which the mean age is around 15 years old. That's a pretty big delay from infancy. Really sets up the need for early life predictors. The table also shows how many of these cases had usable microbiome and metabolic data. So, for instance, only 39 ASD cases had matched microbiome samples, and fewer had cord blood metabolic data. But even with smaller numbers, they're still one of the most comprehensive infant gut brain datasets out there. So, now in panel B, each of these dots represent a child's diagnosis over time, with red showing girls and blue showing boys. So, it's broken down by condition, and what's interesting is how spread out they are. You can see that the dashed vertical line, that's when DSM 4 is transitioned to DSM 5, changed how many of these conditions were clinically defined. So the fact that this study spans both DSM errors add even more weight to the diagnostic data, since it accounts for evolving clinical standards. So you can see that this study was spread out over a good amount of time, even with clinical data changing. And so, I mean, it's really good. It's given a sound background. So what's really key here is that the biological data, like the stool samples and blood metabolites, they were collected way back in infancy before any of these diagnoses were made. That's the strength of this design. It's perspective, meaning that they're looking forward from infancy rather than backwards from diagnosis. So overall, this figure sets the foundation for the study, showing a rich cohort, delayed diagnosis, and a unique opportunity to see if the infant biology can help predict future neurodevelopmental risk. Now let's dive into what I consider personally one of the most compelling results of the paper. So the actual difference in gut microbial species observed between infants who later developed a neurodevelopmental disorder and those who did not. So this is based on figure 4, which breaks down the microbial shifts in the ND group, which is 111 children with future ND diagnosis, compared to the 1,400 neurotypical controls. So in panel A, we see a species like Bacteroides uniformis, and we see Roseburia. And we also see Bacterium, and we see that they're more significant in healthy controls, indicating that they may play a protective role in early life immune and neural development. On the other hand, ND cases had relatively lower abundance of these beneficial taxa. This is actually thought to be from the short chain fatty acid production. So this is compounds that shape the gut brain axis and inflammation. To ensure that these differences weren't due to outside influences like birth mode, geography, diet, or infection history, the researchers use a technique called propensity score, which matches that are shown in panel C and D. It allows them to match ND cases and controls based on potential confounders. Even after this matching, key microbial differences persisted. This means the findings are robust and not even just artifacts of environment or parental factors. Panel E finally explores researchers' subtype specific microbiome patterns. This includes differences between ADHD, ASD, and other ND, and it also identifies signature microbial ASVs using random forest methods. Overall, this figure shows that the gut microbiome at 1 year of age is already stratified in a way that correlates with neurodevelopmental trajectories years later. All right, so here on this slide, we're shifting focus from microbial composition to real-world symptoms that infants and toddlers were experiencing, and how those symptoms turn out to be predictors of later diagnosis. So here on this slide, I want to focus on panel C from figure 2. We see the odds ratio, and I feel like this is where the findings really gain weight. So to transition a bit from the focus of microbial composition to actually real-world symptoms that infants and toddlers were experiencing, and also how these symptoms turn out to be powerful. I want to just highlight this panel. So the study found that children were one or more symptoms in these clusters and had statistically significant increased likelihood of developing a neurodevelopmental disorder later on. So symptoms like constipation, poor sleep, or frequent anxiety which might be brushed off as a normal childhood issue, were actually high risk flags. This is critical because it adds another step in the timeline. We go from microbial imbalance, early symptoms, later in the diagnosis. I want to pull four key takeaways from the symptom analysis. First, we actually learned that symptoms don't happen randomly. They happen from predictive clusters, and by grouping these symptoms, the study revealed patterns that link back to neurodevelopmental outcomes. Also, these behavior flags emerge early before formal diagnosis, before school intervention. Things like anxiety, irritability, and even some of the basics as constipation can be meaningful if they persist to occur together. Third, maybe most of the important facts we get from microbial perspective is that we saw that infants showing more symptoms at 5 years had distinct microbiome patterns at just 1 year of age. That means the connection between gut and brain starts early, maybe earlier than we assumed. Finally, the authors proposed a working model that could help shape the future of research. It is the gut brain behavior pathway, where changes in the infant microbiome don't just reflect what's happening in the brain, but may actively shape it. That gives us a huge opportunity, not just for early screening, but potentially for early intervention. Here we're looking at the first half of the cord blood metabolite comparisons between children later diagnosed with ASD and matched controls. So this data was from cord serum samples collected at birth. Each box plot compares metabolite levels. So controls are in green and ASD cases are in purple. So I want to walk a little bit through panels A through F. So panel A shows a slightly lower lethionine level in ASD group, and lethionine I meant is a non-protein amino acid, and its depletion may point to disrupted sulfur amino acid metabolism, a pathway that's often linked with neurodevelopmental disorders. Panel B shows an elevated IPPA. This stands for isopentyl pyrophosphate. This is a metabolite involved in isoprenoid biosynthesis. This can influence immune signaling and inflammation. Panel C is showing zoluzanone levels. This is higher in ASD groups. So while not widely studied, it may reflect plant-derived metabolite exposure or a marker of oxidative stress. Panel D shows increased levels of 4-cresol methyl hydroxyl 4 isopropylbenzene metabolite. This metabolite has been implicated in neurotoxicity and gut microbiome interactions. In panel E, we have the CT61, which is a long fatty acid chain. Essentially, it's a metabolite. It's significantly lower in the ASD groups. This may suggest early deficits in lipid signaling or energy metabolism. Finally, panel F shows reduced dodecanoic acid or lauric acid, a medium chain fatty acid. This is involved in antimicrobial activity and mitochondrial function. These six panels point to multiple disruptions in metabolic pathways present at birth. So this includes amino acid metabolism, lipid metabolism, and microbial fermentation, all of which is known to contribute to early neurodevelopmental risk. So to continue the data from figure 3, which is an amazing figure, this slide presents panels G through L. Panels G through L are the remainder of the cord blood metabolites, and panel L highlights the key findings in the stool samples at 1 year old. So panel G is the palmitic acid. It's another saturated fatty acid. It's lower in ASD, which aligns with the potential lipid metabolism disruptions. Panel H is octonic acid. This is reduced in ASD as well, which may affect energy production, given its role in the fatty acid oxidation. We have panel I. I'm just gonna call it TUDCA. So basically, this TUDCA is a bile acid with neuroprotective properties. So this is higher in ASD. They reflect alterations in bile acid metabolism, often influenced by gut microbiome. And then we have panel J. We have the UDCA. This is also slightly higher in ASD. Both TUDCA and UDCA, they have been studied neurodegenerative conditions, so this may be relevant. Panel K is the oleic acid. That's a major monosaturated fat. It's lower in ASD cases. This could be tied to early disruptions in membrane lipid composition or brain development. Panel L, it presents only stool-based data. So it shows that at 1 year old, who later developed ASD had significantly higher levels of palmitic acid in their stool. This might indicate either dietary differences or early microbial processing abnormalities. So together, these panels reinforce that both cord blood and gut-based metabolic markers reflect early biological differences in children who go on to develop ASD. So this provides a potential window for early screening and intervention. I wanted to include this volcano plot. It illustrates the differential abundance of microbial species in the first year of life between children who later develop autism and their match control. So each dot represents a species, and the x-axis shows the log two-fold change, while the y-axis shows the negative log 10 of the p-value. So the red dots indicate statistically significant species that pass both thresholds, meaning they were significantly enriched in either ASD group or the control group. Notably, the species like Streptococcus and Rumnicoccus, they're both abundant in these controls. So this suggests, like we were saying before, a potential protective association. So while other taxa enriched in ASD children could indicate microbial signatures of neurodevelopmental risk, these microbial patterns in infancy may act actually as early biosignatures, potentially modifiable before early clinical symptoms. And also, I wanted to show one of the forest plots I was talking about earlier. So this shows the adjusted odds ratios for variables associated with future neurodevelopmental disorders. So this includes ASD, ADHD, and speech disorders. So data integrates multiple domains like pregnancy exposures, parental education, and microbiome data. On the diamonds represent the odds ratio with horizontal lines indicating the 95% confidence intervals. So we can see that maternal smoking and lower education levels are associated with significantly increased risk of ND development. On the microbial side, we see that the Akkermansia levels were also a notable predictor, aligning with the earlier findings on microbial depletion in ND cases. So this figure powerfully highlights that neurodevelopmental risk is multifactorial, combining both biological and environmental contributions from early in life. As part of the study, the authors examined whether host genetics, specifically HLA genotypes, contribute to differences in ND risk. So this is all coming from Figure 5, but I just wanted to break it down a little bit. So what they found were the striking DQ8 homozygous genotypes were enriched in infants who later developed NDs, especially autism spectrum disorder. So this gene is already known to influence immune system development, but here is correlated with lower microbial diversity in the gut. So infants with the DR4 DQ8 had fewer bacterial species overall and reduced community evenness. Beyond diversity, certain key protective microbes were depleted, particularly two main ones that I was saying before, the Akkermansia and the Coprococcus. So these taxa play important roles in maintaining the gut barrier and producing short chain fatty acids that modulate inflammation and brain development. So functionally, the microbiomes of these infants showed suppressed mutation pathways and lower biosynthetic potential, suggesting that the genetic background may constrain the gut's ability to mature and protect the host. They reinforce that idea that genetic susceptibility and microbial composition are intertwined. These findings reinforce that the idea that genetic susceptibility and microbial composition are intertwined, so this shapes neurodevelopmental trajectories early in life. So now that we looked at the species level and metabolomic differences, let's talk about the function, what these microbes are actually doing that may influence NDs. So this slide focuses on three important taxa identified in the BIS cohort. In panel D, we have the Akkermansia and also Akkermansia muciniphila. This is associated in panel E, and then we have the Coprococcus in panel F. So starting with the Akkermansia, we see a functional pathway, basically this enrichment in infants who did not go on to develop an ND. So these pathways include the mucin degradation and amino acid biosynthesis, which we know is both important to our gut barrier maintenance and systematic inflammation control. Zooming in on Akkermansia muciniphilia, this is a species level analysis. It shows a consistent pattern. It's also known for its role in enhancing our epithelial integrity, and this reduces gut permeability, both which are crucial for preventing inflammatory signaling that can affect the developmental brain. Lastly, we have the Coprococcus, which has been previously linked to happiness and dopamine metabolism. So it shows a reduced abundance and this altered functional activity in the ND group. This suggests a possible connection between microbial function and neural behavior regulation. And this can explain some of the behaviors that come with specific NDs. So the takeaway here is that certain microbes, especially those that are tied to gut health and anti-inflammatory functions, aren't just missing, they're functionally silent or underactive in an at-risk infant. This gives us a more mechanistic window, if you must, into how the microbiome might shape early neurodevelopment. I want to talk a little bit about the infant gut microbiome by subtype. And I put this little figure here. I know it's little. I didn't really want to spend too much time on it, but basically it just shows the principal coordinates analysis, called a PCOA plot. It compares the microbial composition among the infants who develop different these. And so each dot basically represents the gut microbiome profile of one infant. The colors indicate the subtypes. And so, yeah, from each group, you can see that there's different clusters in different regions of the space. It reflects unique microbial community structures. So, yeah, supports three main ideas, though. So, first, each ND subtype is linked to unique microbial patterns rather than one shared ND microbiome. Second, despite the differences, there were shared reductions in protective taxa. And third, that these patterns remain significantly after adjusting for early life exposures like diet, delivery mode, and antibiotic use. So this basically suggests that there's a robust link between early microbiome composition and our later ND development. So overall, this analysis highlights how microbial signatures can differ by diagnosis, which could have implications on how we see future screenings or targeted interventions. And then a little bit back on three, I wanted to dive a little deeper in because now since we're talking about the different ND subtypes, slide basically highlights the main findings from the infant stool metabolomics and analysis. So this was examined gut-derived metabolites around 1 year of age. So first, the researchers observed alterations in the lipid metabolism that included significantly higher levels of palmitic acid in the ASD group. We saw before in the slide that there were elevated TUDCA infants who later developed this ASD. So also we saw that the bile acids help regulate the gut barrier integrity and immune signaling pathways. Third, metabolite differences appeared before any clinical symptoms were present. Like, and this shows early metabolic dysregulation that may precede diagnosis. Finally, these results suggest that stool metabolome could have predictive value as a non-invasive biomarker, especially when combined with the microbiome and genetic data to identify infants at higher risk for NDs. Another figure I would like to bring up is figure 7. So it illustrates how the study integrated early life exposures, infections, and microbiome data to better understand their combined impact for ND risk. So panel A, you can see the adjusted odds ratio for several exposures. The authors found that otitis media, which is the middle ear infection during infancy, was significantly associated with higher likelihood of later ND development. So other factors such as maternal smoking during pregnancy and certain microbial patterns also contributed to elevated risk. Moving on to panels B and C, the bar plots show that the proportion of children with and without the Citrobacter and Coprococcus in their gut microbiomes. This taxa was chosen because they were among the most significant associated with infection history. You can see that the Citrobacter was more prevalent among children with otitis and ND, while Coprococcus tended to be reduced in these groups. So panel D and F further breaks down prevalence of these taxa across infants who did or did not have otitis media. So each point represents an individual's child's data. So these distributions highlight that early infections were linked to consistent microbial shifts, especially with children who later developed an ND diagnosis. Overall, this figure supports the idea that environmental exposures do not act in isolation. Instead, they seem to work together in changes in the microbiome to shape early development. In other words, early infections, microbial depletion of protective species, and other exposures may combine to increase vulnerability to NDs. This integrated approach emphasizes the importance of considering multiple factors in combination rather than just studying each one separately. And then one of the last studies I would like to pull in, and start to pull everything together. The researchers wanted to understand not just which microbes were present, but how much of these microbes might actually be influencing metabolic processes relevant in neurodevelopment. This figure shows the results of their integrative analysis, combining microbiome profiles with the metabolic data from infant stool samples. So what they found was that a certain taxa had clear connections to specific metabolites. For instance, the bifidobacterium abundance correlated with the range of lipid metabolites that are important for energy balance and cell membrane function. The Akkermansia showed that associations with several neurotransmitter precursors suggested that lower levels of microbe could translate to fewer building blocks needed for healthy brain development. They also just observed that infants who later developed neurodevelopmental disorders had reductions in metabolites related to shorty chain fat acid production and vitamin biosynthesis. So these patterns point to a less supportive metabolic environment very early in life. Overall, this integrative approach helps us see more complete picture. Overall, this integrative approach helps us see a more complete picture, not just with which microbes were depleted, but also what functional consequences that might have been caused by developing nervous systems. So I wanted to include this slide as a way to just pause and bring everything together, because honestly this paper has so much detail and so many layers that it can be hard to see the big picture at first. So the purpose here is just to simplify and connect. Researchers found that even before any symptoms had showed, babies who had later developed neurodevelopmental disorders had a distinct difference in their gut bacteria. So this wasn't random, it was consistent pattern that were already in place during infancy. And then next, there were clear metabolite changes in both stool and cord blood. So the level of important fats and vitamins were lower, and some compounds that can promote inflammation were higher. So this suggests that the gut environment in the gut was different early on. So also the study showed that how genetics and early exposures work together in a microbiome. So certain immune genes were more likely to have these microbial changes, and infections or smoking made the patterns even stronger. So it wasn't just one factor, it was a combination. Finally, it wasn't only about the microbes that were there, it was also about what they were doing. So, in these infants, the gut microbes were making fewer helpful compounds like vitamins and neurotransmitter precursors. That functional loss could be important to development. To wrap everything up, I do want to highlight a few key points from the author's discussion of what the findings mean. So, first, the study showed evidence of early dysbiosis and inflammation, with gut microbiome and metabolite differences already present in infancy long before any clinical diagnosis. So, these early changes support the idea that inflammatory processes may contribute to neurodevelopmental risk. The results underline that there is not just one factor driving these outcomes. Genetic microbial shifts and environmental exposures all combine to shape risk, summarized in the results. So even after controlling things like diet and antibiotic use, the dysbiosis patterns remain, suggesting that they're inherent biological components. Third, the author found broad functional and metabolic disruptions, depletion of these fatty acids, vitamins, neurotransmitters, how they could affect brain development, immune function, and gut barrier health. So this mechanistic insight may explain how microbes influence neurodevelopment. So finally, the study had some limitations. For example, no detailed neuropsychological testing, and the Swedish birth cohort may not fully represent other populations. Also, only a subset of the cohort had microbiome data. The authors emphasized that more research is needed to understand causality, the persistence of these microbial changes over time, and whether early interventions could help prevent neurodevelopmental disorders. This study, it showed that the gut microbiome and metabolism are already altered in infancy. And, with studies like this and more studies to come, I feel like we can get a great subset that is representative to majority of infants. So some broader implications, these findings suggest that early life gut health could be a window into neurodevelopmental risk, and we still do not know whether dysbiosis causes these outcomes or just reflects them, or whether changing the microbiome early could change the child's developmental trajectory. So in conclusion, the study highlights, everything we just said before, but what I really want to touch on is that the data showed that there were clear microbial and metabolic differences in which later the children develop neurodevelopmental disorders. So these findings are important because they suggest, that the gut could be a window into early developmental risk and potentially target for early interventions. And also knowing these specific bacteria that are beneficial or that are underproducing, we can start, finding ways to introduce these bacteria or suppress certain bacteria in infants and, maybe prevent certain NDs. To help us think more about this, I wanted to share a few discussion questions. My first one says, how might early microbiome screenings be used in pediatric care, and what ethical challenges could that create? Next, which of the microbial or metabolic markers in the study seem most promising as early diagnostic tools? How could future studies test whether modifying the infant microbiome changes neurodevelopmental outcomes? Next question, what role do you think maternal diet and health during pregnancy play compared to genetics and early infections? And lastly, given the limitations of this cohort, Is geography, lack of neuropsychological testing. Generalizable are these findings to other populations? So thank you for listening. I'm looking to hear your thoughts.